about our atomic level to molecular level. Then we went from molecular level to a cell level. And then from the cell level, we take it to a tissue level. And then from tissue to organ, an organ system, to the organism. So what we talked about on Thursday, we talked specifically about the atoms and then the molecules, of which we looked at the different types of bonds that held things together. We had the ionic bonds, and we had our covalent bonds. And we were able to differentiate between the difference between a polar and a non-polar covalent bond. Okay, do you guys remember that? And in our covalent bond, in our polar covalent bond, again, we'll kind of review a little bit because we're going to talk about a specific molecule that's going to be important in our cellular structure. And that is in our polar bonds, polar covalent bonds, we saw unequal sharing of electrons. And in our nonpolar covalent bonds, what we would be able to see is an equal sharing of electrons. And those were our gases. We saw oxygen, we saw CO2, molecules that, for the most part, don't exhibit any electronegativity whatsoever. And the profound molecule that we talked about that had polar covalent bonds was water. And water exhibited hydrogen bonding, which was the third of our bonds that we talked about. And then we talked about our 4 macromolecules. Do any of you guys remember your 4 macromolecules?
Yes.
Okay, name them for me.
Carbohydrates.
Carbohydrates.
Lipids. Proteins.
Lipids, okay. Proteins.
Nucleic acids.
And nucleic acids. So they're the 4 main ones. So when we look at those, what we're going to be looking at today, we're going to be looking at these, some of these molecules. And we're going to take those molecules, we're going to take a look at how they apply in cellular physiology. And going into, kind of as a lead-in, let's take a look at our slides. Now what we're going to do is we're going to talk about our, once this thing loads up, we're going to talk specifically about our plasma membrane. We're going to be specifically zooming out of the molecules that we talked about. We're going to take it now to the cells, which are going to be containing those macromolecules. We're going to see those same things. We're going to see carbohydrates, we're going to see proteins, we're going to see lipids, and we're going to see DNA and RNA. And so now that we have a system of organization, we're going to be able to see how the cell is a basic unit of life, as you learned in biology. And we're going to take that and we're going to look at the makeup of the cell, and we're going to understand the basic physiology of a cell. Ultimately, what we're getting at is that the cell is a factory on its own, and it's able to manufacture what it needs to manufacture. And for the most part, we're going to be talking about the production of proteins, and you'll be able to see that at the end of today. And as a background, when we look at today's lab that's due next Tuesday is going to be that fundamental concept of how we produce proteins. The same proteins that we talked about last Thursday, but you're going to learn how the DNA is tied specifically into them. So what we aim to talk about today, we're going to be talking about the protein carriers and channels embedded within the membranes. We'll talk about a concept of osmosis, which is the movement of water. Tonicity, which is all related to the concentration of a specific molecule in the cell. We'll take a look at the organelles inside the cell that help do what it's supposed to do. And then lastly, we'll talk about DNA replication. So by the end of today, you should have the concepts, so at least of all the cellular stuff. That's the stuff that's going to be on quiz 1. So quiz on Thursday. That's my reminder for you guys. Quiz on Thursday, 6:45. You guys are going to have until about 7:20 to finish it. And then afterwards, we'll get into the lecture. Most of what you're going to be learning, and I will pinpoint the things that are not going to be of utmost importance, but you'll be able to see some of the other topics that we'll talk about as we go into our future lectures. So with that, we're going to go into our first cells. And so what you learned in biology, you had prokaryotic cells, which were your simple unicellular organisms. And now our focus is going to be on eukaryotic cells. And these were more, for lack of better terms, more of your advanced cells. And these cells are going to be defined by having a true nucleus. There is a specific nucleus within that cell that's going to contain the DNA. And we have a nuclear envelope, and you'll be able to see this in our next slide over. here you can see the nucleus. There's a center, just like we saw the nucleus of the atom, you would be able to see the nucleus of the cell. This is the center point. And within that nucleus, you're going to be able to see the DNA that we're going to be talking about. And that DNA is going to contain the blueprints for that entire cell, that factory. And there's a very defined nuclear envelope, meaning there's a boundary to which the nucleus resides within. And also there's a cytoskeleton. And we'll talk about the nucleus, we'll talk about the cytoskeleton. But the cytoskeleton, much like the skeleton in our bodies, provides a support and shape of the actual cell itself. That allows it to have very defined shape as well as some extensions, which we'll talk about in just a little bit. So the cellular anatomy, as you can see, there's a nucleus, there's a cell membrane, which we'll talk about, the cytoplasm, which are all the contents within the cell, of which we'll see cytosol, which is this fluid, and the cytoskeleton, which is going to contain the filaments that are going to give it its structure. And then we'll talk about the organelles, and the organelles of which will have a membrane and will not have a membrane, and we'll talk about those functions later on. Again, high-level overview of the cell. So the first concept I want to get into is the phospholipid, because this is the molecule that I talked about. And if you remember from Thursday, the phospholipid exhibits amphipathic or amphiphilic properties, meaning it loves both water and it hates water. And that is all inherent in the structure of the molecule itself. Here you can see that phospholipid. Remember, this was one of the lipids of which we had a glycerol backbone. Again, you don't need to know the structure, but what I'd like you guys to be familiar with is where the nonpolar and polar heads are. The fatty acid chains. These were long fatty acids with a carboxylic acid that's attached to the glycerol molecule. We had 3 attachment points, 1, 2, 3. The bottom 2 here are going to be attached to the fatty acid chains. And if you remember on Thursday, we mentioned that the fatty acid chains exhibit covalent bonds. But do you remember, were these nonpolar or polar covalent bonds? You guys remember? Nonpolar. Why are they nonpolar? Well, if you take a look at the carbon and you look at the hydrogens around, that electron sharing that happens very equally around. Remember, there's 2 elements that we talked about that are oxygen hungry, electron hungry. They have a tendency to kind of pull the electrons and hold it much longer. Do you guys remember which ones those were?
Oxygen.
Oxygen and?
Phosphorus.
Phosphorus, thank you. So those 2 elements have a tendency to pull those electrons. Here we only really see carbons and hydrogens. Hydrocarbons are covalent, nonpolar covalent bonds, meaning those electrons are shared equally. And therefore, if we were to take a look at water, water doesn't have any point to be attracted to because there's no electronegativity. So this entire region exhibits no electronegativity. Now, on the other end, we would see our polar head. And attached to that 3rd spot on that glycerol molecule is that phosphate group. And here you can see the phosphate ion surrounded by oxygen molecules. Remember those 2 things we talked about, oxygen and phosphorus, are very electron heavy or electron hungry. And so this area exhibits a lot of electronegativity. And therefore water molecules are very attracted to it. So if you remember the structure of the water molecules, the electropositive portions, the hydrogens attached to the water molecules will be attracted to the phosphate ions, or rather the polar heads. So they'll want to gravitate towards the polar head group. So this area would be polar, and this area would be nonpolar, hence the term amphipathic and amphiphilic. They basically mean the same thing. I mentioned earlier amphi, like amphibian, and philic, which is the love of. So the love of both water and non-water. So when we look at the structure of those phospholipids, and you dump them into an aqueous environment, I mean specifically a water environment, what's going to happen is you're going to see these phospholipids. And here we see the structure. And in the book, as well as the diagram below, you would see the polar head would be represented by the large head to it, and then the two fatty acids on it. And you'll see that arrangement take place, where if you dump a bunch of those phospholipids will start to arrange into this formation, where you get this phospholipid layer, and this is called the double phospholipid layer, where you get one layer on the outside, one layer on the inside. Why? Because we have an aqueous water environment on the outside and a water environment on the inside. So therefore, the polar heads would be exposed to the aqueous environments, and the non-polar tails gravitate towards the middle of that bilayer. And this allows for that separation of that internal fluid environment as well as the extracellular fluid environment, of which I'm going to call them ECF on the outside and ICF on the inside. So this serves as the foundation where we're going to be seeing differences between that outside environment versus the inside environment. And if we look at the makeup of cells, this allowed sort of the evolution of cellular biology, where cells can exist in a, I wouldn't say a dangerous environment, but more toxic environment that the Earth had at its earlier phases, as opposed to the inside environment. So you have this barrier that keeps it protected from the outside world. And if we look at the makeup of the fluid from the inside and the outside, you'll note that these are going to be different. Here you can see the intracellular fluid, here you can see the extracellular fluid. And if you're going to look at the two, you're going to note some fundamental differences. And these fundamental differences, you just have to understand and you have to accept. So we're going to draw a cell where we have the ICF and the ECF. There's things that you just have to accept. And this is going to serve the foundation for neurophysiology, so this is going to be extremely important. Taking a look at the inside, the interior or the intracellular fluid, we'll see that the inside happens to be higher in the concentration of potassium. This is my notation for it. Increased concentration of potassium. Okay. Low concentrations of sodium. We've got a lot of organelles, which we'll talk about. We've got a lot of amino acids and proteins. On the outside, we've got the reverse, meaning we've got low potassium, we've got high sodium, and we've got a lot of glucose on the outside. But the real main things that I'd like you guys to be familiar with are these: the concentration of potassium and sodium on the outside versus the inside. This is going to be also a good, another contributor to what we're going to be talking about in our neurophysiology lecture. So these are going to serve as the fundamental differences between these two. And so you can start to see that this is sort of the beginning of homeostasis. We want to make sure that the inside concentration remains high in potassium, and the outside wants to stay high in sodium. And for a cell to exist in homeostasis, for that cell to exist in rest, it needs to make sure that these concentrations are different. And we're going to talk about how that's done a little bit further into our lecture. So now that we have the differences between the two, you can see our phospholipids separating. Let's take a look and zoom in right on that phospholipid bilayer. Here, once again, you can see the hydrophobic fatty acid tails. You can see the polar heads littered both on the intracellular side as well as the extracellular side. And here you can see this is the extracellular fluid. This is the intracellular fluid. So again, as you would expect, outside would be higher sodium concentration, higher potassium concentration on the inside. Now, when you take those phospholipids together, they don't form a rigid structure. In fact, those phospholipids just kind of move around. They swish around quite a bit. And just due to the nature of having that barrier on the inside, aqueous environment on the outsides, these phospholipids literally just swim along. And if you were to kind of take your hand through it, you would eventually kind of swish these things around, they'd move around, but eventually they'd still maintain that barrier, hence the fluid mosaic model that was described in its onset at the very beginning when they described the cell. Now, in combination with it, if you were to see the phospholipids make up about 75%. You don't need to know these numbers, but phospholipids, about 75% of the makeup of that cell membrane. The other components you're going to see, about 20% of that, is going to be the cholesterol. And the cholesterol is theorized to maintain separation. It creates even bigger barrier between these phospholipids. And here you can see cholesterol. Again, one of our lipids with the 4-ring structure. This was specifically a steroid with those 4 rings. And you can clearly see that, 1, 2, 3, 4. And it's embedded within those phospholipids, and it allows for that fluidity in the phospholipid bilayer. Now the other 5% are glycolipids. And these you can see are present. Glycolipids right here are protein, or actually rather, in this case, these are phospholipids with sugar, chains of sugar molecules or glucose molecules embedded at the very top. And they kind of look like antennae because that's exactly what they do. They allow for cell signaling, meaning they give characteristics to the cell itself. What do you mean by characteristics? Well, think about these. You've heard of blood types, right? You guys know your blood types? A, B, or O? Well, these are cell signals that tell you exactly what red blood cells might be comprised of, whether you're a type A, type B. It's a marker to basically tell the human body that these are human cells. In fact, these are specifically my cells. And these glycolipids serve as signaling just to say, don't attack me, so that the immune system doesn't come in and say, I don't recognize you. If the immune system doesn't recognize it, it'll come up and literally will chew it all up. But if you have those markers on your cell, then it would preserve those cells. Kind of the whole concept of transplant, why if you were to get transplanted an organ and they want to match compatibility, what are they looking for? They're looking for cell markers. They want to make sure that those cells closely resemble yours in terms of the cell signaling so that your immune system doesn't come in and attack it. Of course, they can't get 100% match. They might get 90, 80% match. But that's why people that have transplants, does anybody here know that anybody that's had a transplant? Maybe one person? Transplant, people that have had a transplant have to take a lifetime dosage, not a lifetime dosage, but lifetime supply of immunosuppressants, meaning it suppresses their immune system from attacking that organ. Have you ever heard of organ failure or organ rejection? That's exactly what happens, right? These lipids aren't present on those cells, and therefore the immune system comes in and literally just attacks it. Autoimmune system also, or autoimmune disorders work the same way. Those cells that might be produced might come in and they might not see those glycolipids and they'll come in and again attack those cells. So this is our way of doing it. So that's what we've talked about. Again, we've talked about our phospholipids, we've talked about our cholesterol, and we've talked about our glycolipids. The next thing we're also going to look at are proteins. And you can see that some of the proteins span through the entire width of the phospholipid membrane. And then you can see some other proteins that are kind of just embedded on the inside or maybe on the outside of the cell. So we'll talk about these proteins, but you can pretty much determine, or maybe not determine, you can sort of estimate what that function of that protein is based on the location. So by looking at whether the protein spans across or is embedded on one side, you can pretty much know what it might be able to do. And we'll talk about it a little bit those in just a little bit. So the next thing we're going to do is we're going to look at this concept of permeability. And permeability is the movement of molecules through something. While we know that the phospholipid membrane can maintain a separation of molecules from one side, the extracellular fluid from the intracellular fluid, we still need the movement of molecules and the movement of stuff inside and out. And how do we do that? Well, we have to look at the permeability of the membrane. And the permeability can change over time, and I'll talk about how that can get changed. Let's first talk about specifically how things move through. Well, the characteristic of the phospholipid membrane is that it is selectively permeable, meaning it only allows stuff to move through if it has things that allow it to move through. And the first thing we need to look at is the makeup of that molecule, hence why we spent time in understanding our nonpolar versus polar bonds. So looking at our permeability, the first molecules that can move through unhindered would be nonpolar uncharged molecules. So things like gases, we look at oxygen and carbon dioxide. O2, O, double bond O, and then CO2, which would be C, double bond O, double bond O. These don't exhibit any electronegativity. Why? Because the molecules are shared equally. You might be thinking, well, Marlon, you mentioned oxygen. Oxygen happens to be electron hungry, but you get an oxygen molecule on one side, an oxygen molecule on the other side, and therefore there's going to be equal sharing. They're both electron hungry, but these are double covalent bonds which are nonpolar in general. Same goes with CO2. So with that, these gases move in and out fairly easily through the phospholipid membrane. They're small and they pass through. So these are our non-charged molecules. Now ions, on the other hand, things like sodium, things like potassium, they can't move through. Why? Because they're kind of attracted to the polar heads and they can't move past it. So therefore, ions don't cross through the phospholipid membrane. How do they move through? We'll talk about that in just a little bit. Other things, other large molecules, macromolecules, can't fit through. They're just too big. So even though the phospholipid molecules move through, that molecule just can't seem to squeeze right through those contact points. Other things to look at are lipids. Lipids have the tendency to be, lipids are actually nonpolar in nature, so they can slip right through. They're not attracted to the polar heads. They're somewhat attracted to the hydrophobic region, but they'll slip right through. So that is an important concept. Lipids, and then impermeable to ions, so things that are polar in nature. Polar molecules can't cross through, nonpolar molecules can. And so this will be important when we talk about in our endocrine lecture, how do we get things to move over and target the nucleus? So keep that in mind. Then the next thing we need to talk about is water. Water can move through. Even though water exhibits polar covalent bonds, and I did say that polar stuff can't move through, water is the exception. And if we look at water molecules have a tendency to be small. You would think, given the concept that we talked about where water would stick to the polar heads, it's small enough that it can actually fit through. So water is that exception, hence why water is one of those molecules that's widely studied, something that is of profound interest, not on just this Earth, but on our other planets. The last thing I want to talk about on this slide is the concentration gradient. So concentration gradient is exactly what I described earlier, meaning we have a higher concentration of sodium on the outside, we have a lower concentration of sodium on the inside. And whenever you have more of one thing on one side, that's going to give us a gradient. So if I can use an example, if I can imagine that my left side of the room is going to be intracellular fluid, right? So you guys are the inside of the cell, and you guys are on the outside of the cell. And right through the center of this class is that phospholipid membrane. So with that, if I were to take all of you guys, and all of you guys are just random molecules, and we'll just say that you guys are, I don't know, sodium for instance, and I cram you all onto one side of the room, in this case on the extracellular fluid, there would be a tendency to want to diffuse across to the other side. So with this concentration gradient, that is going to be the driving force that will guide which molecules are going to move in which direction. So if I were to take, in this concept on our next slide, the diffusion capacity is tied into the concentration gradient. Let me redact that statement. That's one contributing factor. So diffusion capacity is the likelihood that things spread out. So if you remember that stink bomb example that I talked about on Thursday, I drop a stink bomb on the ground, right? You have all these molecules that have a sulfhydryl group attached to that molecule. It's going to break open, and now you get these molecules that start to diffuse across the room. When it's broken immediately, you might not smell it. But give it about 5 seconds later, all of a sudden you can start to smell it. Why is that? It's exactly tied into the concentration gradient. Within that stink bomb, you have a high concentration of those sulfhydryl molecules. The tendency of those molecules are to disperse and move across until they're equally spaced around the classroom. Something hot works the exact same way. This is the diffusion of heat. If I took a cup of boiling water and I placed it down on the table, eventually heat will start to disperse per the second law of thermodynamics, will disperse out into the room until it starts to heat up and eventually distribute equally. If I were to take all of you guys and cram you all into the corner, you'd all get uncomfortably close to one another. And if I say, okay, now you guys can move around, which way do you think you're going to move? Are all you guys going to move to the opposite corner and cram on the other side? No, you're all going to space out eventually until there's equal enough room. That's exactly what's happening with molecules in an aqueous environment. They have a tendency to kind of spread out. And you can see that on this top right image. You have a bunch of stuff dropped in there. And let me give you even another example, a sugar cube. A sugar cube is exactly this. You drop a sugar cube into water, and eventually it dissolves. They start to diffuse equally into that cup of water until all the molecules are all spread out. So when we talk about the movement, whether it's the movement of heat, whether it's the movement of molecules in an aqueous environment, we have to talk about the diffusion capacity. At what rate do these molecules disperse in an aqueous environment? So let's take a look at the contributing factors to those rates. So using that example again, intracellular fluid, extracellular fluid, right in the middle is our phospholipid membrane. If I have a bunch of you guys, and let's say you guys can move through that phospholipid membrane. If I had a bunch of you guys crammed into one side, let's say the extracellular fluid, the tendency would be exactly what I described here. You guys would want to disperse and move through the phospholipid membrane until you'd eventually equal out. Okay? Assuming that you guys can move through the phospholipid membrane. So the more people I cram to one side, the more likelihood that you're going to move in this direction into the intracellular fluid. If I have all you guys crammed on the intracellular fluid, which direction would you guys want to move? To the extracellular side. If you are both equal on both sides, do you get any movement? Actually, yes you do. The net movement is 0, meaning you get 3 you guys that move to one side, you get 3 that move on the other side. So net movement would be 0. So you guys are, I guess, technically right. There's no net movement. Now, what could I do to speed things up? What happens if I introduce more kinetic energy into this? Meaning, I add temperature in the form of heat. So if I start heating up molecules, what does heat do to a molecule? Kind of speeds it up, right? You're basically just a bunch of molecules that are just jiggling around in space. And if I start adding heat to it, those molecules move around faster. If I start making the solution even colder, eventually you start to slow down. You have reduced kinetic energy, and therefore less likely to move. And if you can, and kind of expanding on when we talked about our 3 laws of thermodynamics, the third one was absolute 0. And that's where I was talking about if you could get a solution down to absolute 0 temperature, that would basically mean the molecule stops moving. We can't get to that point just yet. So if I increase the temperature, I increase the diffusion capacity. Now, and steepness of gradient is going to be an increase. Now, mass, if I make the molecules bigger, right? If I make the molecules bigger, do you think that would speed up or slow down the rate of capacity? Slow down. This is simple physics. The bigger the object, the slower it'll move. The less likely it'll move across the phospholipid membrane. So this decreases. Now the surface area of the membrane is the amount of space that you guys could diffuse across the membrane. So if I were to say, your space to move across is only this big, and all you guys can only filter through that space, do you think that's going to speed up or slow down diffusion capacity? Slow it down. But if I were to increase the surface area, I'm going to make it the entire length of this room. That should speed it up, because you guys have more areas to move through. So the surface area of the membrane increases the diffusion capacity. Now diffusion distance is the last one. This way I'd be talking about not the surface area, but the width, how long it would take. So the phospholipid membrane is like this gelatinous substance. So if I were to take that width and make it even wider, would that slow down or speed up?
Slow down.
Slow down. So the diffusion distance increases. So now you might be thinking, well, in what cases would you be able to see diffusion distance? Well, things like edema, and we'll talk about this a little bit more, but the buildup of fluid around the cell itself could start to thicken the width of that.
That would decrease it, right?
The diffusion distance?
Yeah.
So diffusion distance would decrease the diffusion capacity. If I were to make this thicker, that would mean that molecules would move slower. So you would be moving through that phospholipid membrane and then trying to move through this extra layer on the outside, eventually to make it to the inside or vice versa. So it's a further distance you'd have to travel through that barrier.
Where did that arrow?
Would it be
Yeah.
slow down?
Oh yeah, that's right. It would be right if I were upside down, so that's not. So. There you go. Good catch.
Okay.
I would like a tricky thing for them.
I want to trick you guys. So that, to recap, you would see the ones that would slow down would be the mass, the size of the molecule, and then the diffusion distance, the distance you have to travel. But the contributing factor here, especially in a physiological environment, is going to be this one, the steepness of the gradient. And you'll be able to see that when we look at our, which I call the YouTube example in just a little bit. So now that we've talked about molecules moving through, and we talked about, in the last slide over, we talked about selectively permeable membrane, meaning we've got this barrier and we have molecules that can either move through or not move through, where the most part, non-polar molecules can move through. Polar molecules, such as charged molecules and ions, have a tendency to not be able to move through. Let's talk about how they can move through. Almost everything can move through with certain mechanisms. And we're going to take a look at two different types of diffusion. We have passive diffusion, and then a couple slides down, I have active diffusion, or in this case, active transport. And the main difference between these two is it requires, well, at least active requires ATP. If something's passive, it just happens. If something is active, it requires energy, and that energy comes in the form of ATP. Remember, that's what we talked about at 9:35 on Thursday night. That was specifically ATP. It was adenosine triphosphate. That was that molecule that forms that energy currency. Now, passive diffusion, we have two types. We have either simple or facilitated. So one way I can kind of draw this out, we'll talk about passive, and we'll talk about active. Passive can be divided into simple versus facilitated. Simple diffusion is exactly the mechanism that we were talking about, where things such as nonpolar molecules that have a tendency to be hydrophobic can just move across. So non-charged molecules will move across through the phospholipid membrane. It's simple diffusion. You move through the phospholipid membrane. The other is facilitated. What do I mean by facilitated? Well, think of what the term facilitate. Facilitate means something to help. Something to help that molecule move through. So here you can see, these are our hydrophilic molecules. So things like, things like our charged molecules and ions, for instance. They can only move through if they have something that helps it. And in those cases would be channels. Channels, think of any channel that allows boats to move through bodies of water. I failed geography. I didn't do so well in geography, so I can't really think of where there are channels in the world, but they allow boats to move through. Boats can't move through land unless there's a channel that allows them to move across. That's exactly what these things are. In this case, of the analogy of the boat, I'd be talking about charged ions. Now, carriers, on the other hand, are things that allow larger molecules to move across, and I'll have an example for that in the next slide over. But before I get to this slide, I do have to talk about what these helpers are. So what facilitates facilitated passive diffusion? These are those proteins. And let me go back to an earlier slide where I talked about the location of these proteins among the phospholipid bilayer. We have transmembrane, which span across the entire width of the phospholipid membrane, and we have peripheral ones where they just land on either the extracellular side or the intracellular side. And this I pointed out in our other slide. Here, this would be an example of a transmembrane or integral protein. The book uses integral protein. Transmembrane is the same thing. It's trans the entire membrane. And in these cases, transmembrane, if you look at the transmembrane proteins, and this is an image I've been dying to change forever because it's so blurry. But if you look at the protein, you can tell the function of that protein as a facilitator based on its location. So if the protein spans both the intracellular side and the extracellular side, it must mean it's doing something on both sides. And that's exactly what these ion channels do. They allow specific ions to cross through the membrane. And these ion channels are attuned to the specific channel, or to the specific ion. So we'll talk about sodium channels, we'll talk about potassium channels, and they only allow that ion to move through. Now, carrier. These are for the other facilitated diffusion. Carrier are transport proteins that allow molecules to move across, but they literally have to carry it from one side to the other. And so the term that you're most likely, I know most likely you're going to be able to see this, ligand. The term ligand is a molecule that binds to a carrier. So be familiar with that term because we'll be throwing this term quite a bit, ligand. A molecule that binds to the carrier. And let me give you the example of a sugar molecule on the next slide. In order to get glucose into the cell, it's a large molecule. Now, it tends to be polar, so it can't move across the phospholipid membrane on its own. So this transporter allows glucose. Remember, glucose is high on the outside, and we need to get it into the cell in order to generate ATP. If you're wondering how, we'll talk about that on Thursday. We need to get glucose into the cell. We've got a glucose transporter that binds glucose onto the active site on the extracellular side. Once it binds through, we're going to get this configurational change that allows glucose to spit it out to the intracellular side. So here, because we can see the gradient exists from the high concentration of glucose on the outside to the low concentration of glucose on the inside, these carriers allow that movement through one side to the other. So this is one of the things that you're going to be able to see. So you might be wondering, well, does that mean we're going to take all the glucose on the outside of the cell and shuttle it inside? Well, there's a way to modulate this activity. And to kind of give you an example, we kind of talked about it on Thursday. There's a way to stop this glucose transporter. We don't want to take all the glucose that's available outside and shuttle it in. We only need to take what we need in. And the way we can do that is we use something. So if this is our active site, and I know I'm using these terms kind of loosely because this isn't necessarily an enzyme. There are other sites, kind of like allosteric sites, that can come in and activate this transporter. And if you're wondering what that thing that would bind to this thing right here, well, one of the things that is a common thing to activate these glucose transporters is insulin. So if I were to use that example earlier, this is a phospholipid membrane, and I'm trying to carry all you guys. You guys are glucose molecules. I'm wanting to move you guys one by one. Something needs to tell me to stop. No, we've got too much glucose on the inside. So if insulin makes its way and attaches to me, that's when I turn on, and I can now move molecules in. And eventually, when that insulin is taken off, then I stop. Okay? So insulin is a common thing, and the direction of which glucose moves, and nothing you really need to know for now, is really dependent on which cell we're talking about. Right now, we're just talking about the generic cell. But you'll be able to see that there's different types of glucose transporters. And this example that I'm using, again, not something you need to know, but this is GLUT4. This is one of the glucose transporters that is activated by insulin that allows glucose to move into the cell to be used. And these are common in muscle cells. You would see these typically where if a muscle is active, it needs sugar in order to start generating energy for the cell. There are also glucose transporters in the liver, in the kidney, small intestine, for instance, another thing where we're going to have extracellular fluid and we want to get glucose into the bloodstream. So a different type of transporter. And what would be the main differences between the glucose transporters? Is just the makeup of it, the structure of it, the way it's folded. Again, something that we'll talk about a little bit later on today, and you'll get an idea of how we encode the blueprints, the primary sequence for this protein that will lend itself to the secondary sequence, that will lend itself to the tertiary sequence, and then putting all those together, our quaternary structure for putting that protein together. Again, tying what we talked about on Thursday. So if you were to think about ways to increase, and let me tie this into, for instance, exercise. Okay. If we look at the generic muscle cell, and we know that sugar molecules, or glucose molecules, are used to generate ATP on the inside of the cell. We would think that we would want to now be able to shuttle glucose into the cell a little bit faster. If you're doing things like resistance training, if you're doing activities that require a substrate in order to generate ATP, we need to get energy, or we need to get a molecule, glucose, faster into the cell. So looking at these mechanisms, what ways can we increase the amount of glucose moving in? Well, one of the ways, we can increase the number of carriers. So increase the number of carriers. The more carriers we have, the faster we can shuttle glucose in. Can you guys think of another example? Might take a second, but kind of chew through it.
Increase insulin.
You can increase insulin. That's another response. So increase insulin, your target molecule. We can reduce the thickness of the cell membrane, even though I can't really think of a relevant example of where that would take place. Can I increase the temperature of the muscle cell? How do we do that? When you're exercising, are you cold all the time? No, you generate heat. You generate heat, we increase the kinetic energy. That allows glucose to move faster. We get those carriers to move faster. We're not doing anything in the mass, that glucose is going to remain the same. And then the other, the steepness of the gradient. We get glucose more readily available on the outside. So therefore, if we're increasing diffusion capacity through these channels, we get more glucose into the cell. You get more glucose into the cell, you can generate energy efficiently. And again, we'll talk about the mechanism of that in 2 days. With that being said, what questions do we have? So last thing I want to look at for our integral proteins are the peripheral proteins. We've talked about the transmembrane proteins that allow movement in and out of the cell. Now the peripheral enzymes or peripheral proteins are things that are bound to either the intracellular side or the extracellular side of the phospholipid membrane. So we have either enzymes of which we'll talk about, that perform actions on that side of the cell. Did you have a question? The other ones that we're going to talk about are receptors, where receptors can exist on the outside of the cell, and they receive a signal of some sort, and that signal can be a hormone. Again, we'll talk more about that in our endocrine lecture, things that allow hormones to bind to the cell itself. So let's take a look at, we've talked about passive diffusion. Let's talk about active transport. And the key thing that describes active transport is the term active, meaning it requires ATP. It uses energy in order to move ions against their concentration gradient. Why would we do that? Well, we're going to learn that there is sodium on the inside. Well, we already know that. There's sodium on the inside of the cell. How do we get, if we start to see an increase in the sodium on the inside, and again, we'll talk about that next week when we get into our neurophysiology lecture. If sodium starts to come on the inside, how do we get it back out? We have to pump it across, and we're moving against a concentration gradient. So if the concentration gradient for sodium is towards the inside, and potassium, concentration gradient is going on the outside, in this case, this would be the intracellular fluid, this would be the extracellular fluid. We are pumping potassium out of the cell. So against the concentration gradient. And we're moving, sodium is, we're moving sodium against its concentration gradient, and we're moving potassium against its concentration into the cell. And that requires energy. If you were to look at levees, they basically work the same way. Levees where you have water, you have a high level of water, and you've got a low level of water, and you're trying to get water back onto the higher side. You've got to use electricity to pump that water against. If I wanted to go the other way, well, you can just open up channels and just allow water to move in. This way, you require energy to pump it across. And the best example of that is the sodium-potassium pump. Again, we'll talk about this next week. But that sodium-potassium pump has a third attachment point, which is the ATPase. And the ATP is used as the energy to pump them across the gradient. So whatever sodium's left over gets pumped across, and whatever potassium is on the outside gets pumped back in. So now we've taken a look at different types of channels. We've looked at different types of channels and carriers. The reason why there's a lot of, at least for a good chunk of studying pathophysiology, and when you guys do get into pathophysiology, you'll start to look at pumps, you'll start to look at the dysfunction of pumps and dysfunction of channels as a setup for why diseases take place. And there's a whole term that's been used, that's been thrown around quite a bit, called channelopathies. So one of the things they want to do is study the dysfunction of that particular pump or channel as a major cause of a particular disease. To give you an example would be seizures. And when they look at seizures, they note that there's a dysfunction of these potassium channels. If you look at cystic fibrosis, there are chlorine channels. And I've talked about sodium and potassium, but there is chlorine also. There's a bunch of other ions. Chlorine channels are dysfunctional with people with cystic fibrosis. And therefore they get a buildup of chlorine, and that disrupts the electrical balance. And what do I mean by electrical balance? That's something we'll visit next week. But to give you an idea behind all this is to look at what happens with these ions. So if I were to draw our bilayer, and I have, well, let's just look at sodium, for instance. Well, actually, let's look at potassium. Potassium is high on the inside. We have low potassium on the outside. So if I get a bunch of potassium ions, potassium has a positive charge. If I have more positive ions on the inside of the cell, in respect to the outside of the cell, that makes this side more positive, makes this side more negative. And we'll explain this concept in much more detail next week. And that lends itself to a certain electrical difference, which we describe as a voltage between the two. Again, if you're not getting that now, that's okay, because we'll revisit this next week. So, in preparation for our neurophysiology lecture, I'd like you guys to be familiar with this voltage gating that should take place, because I've seen people get stuck on this whole idea. And in these ion channels, specifically a sodium channel, it requires a certain electrical difference from both sides in order for it to operate. And we're going to talk about the changes in the voltage in that phospholipid membrane. These voltage-gated sodium ion channels require the differences in charges that allow it to open up. So looking at the makeup of the sodium ion channel, and I'm just using this example here so that you can understand the fundamental concepts of charges and what opens these things up. We learned about insulin opening up glucose carriers. Here, sodium ion channels are activated by voltages. So on top, you'll see activation gates. This one's another gate called the inactivation gate. And at rest, these channels are closed. And once we get to a certain voltage, meaning negative on the outside and positive on the inside, and eventually there's going to be a certain change where you're going to open up that activation gate. And as long as that inactivation gate is open, you're going to get ions to move through. And eventually, once you get the ions moving through, that's going to cause a change in the voltage that's going to result in this inactivation gate closing, which is going to prevent things from moving through. That might not make sense now. I can tell by some of your faces you're looking at it, you're like, what is he talking about? That's okay. We're going to revisit this when we talk about that physiology, the neurophysiology lecture. We're going to be talking about a specific type of cell. Right now we're talking about generic cell. We'll be talking about a nerve cell that's going to exhibit these properties that's going to allow for the voltages to change. So stay tuned for that. So with that, let me see, Okay, let me finish this portion up, and we'll take a break before we get to this one. So we've talked about the movement of molecules through a cell. Now we have to talk about how things move in and out of the cell. How do we get things in and out that are much bigger? And here we have to employ what are called vesicles. Vesicles, just like the term, I guess, somewhat similar to vessels, are basically sacs made from the cell membrane. So yes, they contain phospholipids. And the way we would do that, and I'm going to show you a slide on the next slide over, showing you an example of how we get things into a cell. So what we're looking at here is a microbe, and this is a fairly large, obviously not something that should exist in the human body. This is a cell that needs to get broken down. And what cells do is that they can take in things from the outside world, things that move into the cell. And this is what we call endocytosis. And if you look at the term endo means into. So things moving into the cell. The exact opposite is exocytosis, things that move out of the cell. This example is an example of things into, so this is endocytosis. And endocytosis can get broken down into two terms, phagocytosis or pinocytosis. And the fundamental difference between these two, phago meaning to eat, pino meaning to drink. So phago is to eat large particles, pino is to drink large amounts of extracellular fluid. So whether we're talking about particles or we're talking about fluid. And in this case, this is exactly what phagocytosis is. We get a microbe that comes in and attaches to the receptors somewhere on that membrane. So it comes in and attaches to. And then we get, and you don't have to know about these pseudopods, but these basically will wrap around, and they wrap, the whole cell wraps around that microbe. And, no, don't restart. We get the cell membrane that wraps around, and what it does is it contains that microbe on the inside, so it can't really do anything. And it's that cell membrane wraps around, you get this phagosome that's wrapped, you get the microbe that's wrapped around by that phagosome, and then you have these lysosomes, which I'll talk about later on. These are digestive enzymes that will come in and fuse together with that phagosome to come in and chop that microbe into a bunch of pieces. This is our way of a cell saying, I don't recognize this thing that doesn't exist. Let's come in and swallow it whole, break it down, and now you've got all the components of that cell. It's a way for cells to kind of eat something out. Pinocytosis, on the other hand, is just basically a bunch of fluid. If there's a fluid that's like high in glucose, or a bunch of glucose molecules in that area, pinocytosis, I don't even use the word glucose. Let's say just say some ion that comes in, and we want to take a good amount of it in. That can get wrapped up and get swallowed in. That's pinocytosis. Exocytosis, we'll learn later on. These are ways we can get things like proteins to make its way out of the cell. So we'll use that example later. What questions do we have?
So basically, this is how they kill cells that are foreign to the body?
Exactly it. Exactly. It's much more complicated than this, but the whole...
Does that have to do with the lymphatic system?
Yeah, there's immune cells in the lymphatic system. If you're looking at specifically lymph nodes, there's ways to do that. But the immune system, and one of the things I really wanted to get into is immunology, where we talk about the immune system and the cells that are involved in eating things up, things that don't exist. Okay. This is essentially how we get rid of foreign bodies in our system. It's through this process of phagocytosis. We come in, chop it up, and we use those parts. Any other questions? Yes?
So, the ion that you're saying there, that's a part of phagocytosis, it sort of like drinks it in? Is that what you're saying?
That's all it is. It's just ways for cells to take in a good amount of fluid to bring it into. That's all it is. My computer? Someone calling my computer? Okay, just ignore that. I'm going to ignore that. And let's take a break, shall we? Let's do you want to guys do a 5-minute break, 10-minute break? 10-minute break, sounds good.